All right, students, this is a recording for Biology 1407, Chapter 39, the respiratory system. This will be the last of the chapters that's included in this unit for this third exam. That is the nutrition or digestive systems, the endocrine system, the circulatory system, and the respiratory system. All right, so let's go ahead and begin. I'm going to start by covering one bit of material that we didn't get covered in the last recording properly. So this is one item that, looking back over the study guide, I realized I hadn't lectured on. And this has, the question was in your study guide, it asks you to know about hydrostatic pressure versus osmotic pressure in a capillary. So the idea here is that blood, coming from the heart via the arterial system, leaves an artery and goes into a capillary, and the pressure I want y'all to know is about 40 millimeters of mercury right here. So that's the hydrostatic pressure, the same sort of pressure that makes water come out of the faucet. And that pressure drops as we go through the capillary right here. So by the time we get to the venous end of the capillary, it's about 20 millimeters of mercury. 40 going in, about 20 millimeters of mercury going out. The capillaries are permeable, kind of like a soaker hose, those garden hoses that leak water as little droplets off of what appears to be a solid surface, a foamy surface. That's kind of the way the capillaries work. So that hydrostatic pressure squeezes water out of the capillaries, and that is the source of the interstitial fluid in your body tissues. Where does that liquid come from? Well, it's literally plasma leaking out of the capillaries because of hydrostatic pressure. Now, at the same time, the material in your blood, the liquid in your blood, has a higher osmotic potential than the tissues in than the fluid in the surrounding tissues right here. So there's always a net draw osmotically of water back into the capillaries right here. But that osmotic pressure, as it says right here in the text, is around 25 millimeters of mercury drawing water in. But the hydrostatic pressure at the beginning is about 40 millimeters of mercury pushing out. So 40 beats 25, so the net pressure out is 15 millimeters of mercury. So water leaves the capillaries. But by the time we get to this end down here, with that loss of water from the capillaries and the reduction in pressure because of that, and because of the distance, the effective pressure reduction over distance, the hydrostatic pressure squeezing water out is around 20 millimeters of mercury, but the osmotic pressure pulling water back in is 25 or so millimeters per mercury, maybe even a bit higher. And so the net flow is of water back into the capillaries at the end right here. So nutrient and hormone and oxygen and water and salt-rich liquid squeezes out into tissues right here, delivering directly to the surfaces of cells. And then that same fluid, now full of CO2, ammonia byproducts from cellular respiration and protein metabolism, and hormones produced, if this is hormonal tissue, signal molecules, whatever, that liquid is all drawn back into the capillaries right here and returned to circulation. We don't lose cells normally in here. We don't lose the large molecules like the colloidal molecules, like the plasma proteins and all of that sort of stuff. But monomers of nutrients and gases and salts certainly leak out with this liquid right here and perfuse the tissues around here. So that's how capillaries work in you. All right. Now let's go ahead and talk about gas exchange and respiratory systems in animals in general, and then in you in particular. Okay, so most eukaryotic cells require oxygen for use in efficient production of energy. That's most, not all. There are a few very rare eukaryotic organisms that don't even have mitochondria, so they don't need oxygen. And then a lot of your cells can work anaerobically as well and don't need oxygen. Some of your cells, adipose tissue cells, for instance, don't really need a lot of oxygen and typically have a very small number of mitochondria, if any at all sometimes. So there's variation in that answer, but as a rule, a eukaryotic organism has mitochondria and therefore requires oxygen. You all know that lots of bacterial cells are anaerobic. They don't have mitochondria. They are not structurally similar to mitochondria. They don't need oxygen. And as we say right here, a major challenge for a big multicellular organism like the sea turtle right here is successfully getting oxygen and nutrients, to the deep tissues scattered around all in its body. This may seem like a small neck right here, but from a biological diffusion perspective, it's an incredibly huge structure right here. And any exchange on the surface right here, which reptiles don't do, but let's just pretend it does for a second, is not going to make it to cells really deep within this neck right here. 6 cells in is just going to be the tiniest little bit of distance through a teeny bit of the skin right here, let alone the entire thickness of the neck. So, there's got to be some mechanisms for improving that diffusion of nutrients and gases and whatnot to those internal tissues. And the same thing for getting rid of carbon dioxide and other waste products, metabolic waste products. All right. So this is looking at alveoli, which are features of lungs in terrestrial vertebrates. But and we'll go back and do some general stuff in a minute. But just some generalizations right here when we're talking about gas exchange. So, and this is true for all gas exchange everywhere. But in vertebrates, the gases diffuse, into an aqueous layer of liquid, that's covering the epithelial cells that line the respiratory organs. So if you look inside your lungs at the alveoli, which are the tiny little, gas exchange sacs, the termini of the, that's the ultimate business end of your lungs, that alveolus is lined by epithelium, and that epithelium is covered, in a mucousy, watery sort of layer right there. And the gases dissolve into those liquids, of course. And remember, gases only move by diffusion, as far as, moving from one chemical place to another or across the cell membrane or anything like that. All of that movement is by diffusion, right? So when we're looking inside the lungs, the O2 is very O2 level. The concentration of oxygen is high and the gas is in the alveoli, and it moves down its concentration gradient into the blood of the alveolus. At the same time, the CO2 concentration in blood is greater than that in the air spaces of the alveoli, or even the liquid covering that epithelium, and CO2 leaves the blood and moves into the alveoli. This will happen as long as CO2 in the atmosphere is lower than CO2 in the blood, which is the normal state of things. If you go into a very high CO2 concentration environment, this starts backing up in your body tissues, bad things happen, and if that backup is great enough, it can be fatal. The opposite occurs in tissues, right? So O2 that's concentrated in blood, when you move through tissues, if that tissue has a lower concentration of O2, which is almost always the case, the O2 moves out of the blood and into those interstitial fluids. And CO2 leaves those interstitial fluids and moves into the blood because it has a lower concentration than tissues. The diffusion gradient for CO2 when you're in good health and in the appropriate environment is from body tissues to interstitial fluid to blood to the alveoli of the lungs and then out to the atmosphere. The pressure gradient for oxygen under normal conditions, if you're healthy and the environment is as we expect it to be, there should be the greatest amount of oxygen out in the atmosphere. Drawn into the lungs drops a little bit because we have some stale in there, but it's still higher than the blood, so it moves on into the blood. The oxygen concentration of blood is higher than it is in body tissues, so it's going to move into interstitial fluids and on into the cells of your body tissues. All right, a little bit about gas exchange. I'm not going to require you to memorize this formula right here or do manipulations of it. Unfortunately, I wish we could, but we don't have time. But you do need to be familiar with the idea, all right? So the rate of diffusion of a gas or a nutrient is, the product of a number of different factors. And so, this is one of those physics-like equations right here. And the key things are the diffusion constant, which is specific to the material, sugar versus, glucose versus sodium versus potassium versus a big protein. They all have a distinct D value here, this diffusion constant, okay? A bunch of things go into it, but it's a constant for a particular atom or chemical or molecule. A, the area which that diffusion has to occur. And then that is also, so that diffusion constant and the area multiplied by the pressure difference between the two sides, and then all of that divided over the distance which diffusion has to occur. So from one cell to another, from 4 cells, 5 cells, greater physical distance, whatever that is. So those are a bunch of factors. Again, you don't have to memorize this, but that's what this thing means. So this times this divided by that is equal to that. So how can we as organisms increase this R value? That's the real question. And so we do it by changing these numbers right here. So basically this term up here, the upper term gets bigger and the lower term gets smaller. So if we can reduce distance, we increase diffusion rates. If we can increase the pressure difference between beginning and end, we can increase diffusion rates. If we can, increase the area in which diffusion occurs, we can increase diffusion rate. So increasing area, increasing the pressure difference, all increase rate of diffusion. Decreasing the distance that you have to have diffusion occur across also increases R. So making this smaller or making either of these bigger, we can do. This is fixed based on the gas or the molecule. So let's simplify that idea here. Evolutionary changes have occurred to optimize the rate of diffusion of R. And the reason we want to increase or optimize the rate of diffusion of R is so that we can be bigger, we can move faster, we can do harder things. Those are the, to be larger and, and stronger and faster and all those things requires increasing R. All right. So how can we do that? We can increase surface area, and we know how that happens. Highly branched and very, very folded exchange surfaces. Your lungs actually have a huge amount of surface area packed into a relatively small organ. You'll hear numbers, like the surface area of a football field, for your gas exchange surfaces. So that's an example of that. Same thing in the circulatory system with all the folds on the intestinal surfaces and the folded intestines. Decrease the distance D. How do we decrease the distance that diffusion has to occur, from A to B? Well, that's what circulatory systems and body cavities are for, is to bring that distance from the outside to the inside down virtually by having this high-speed fluid moving around all over the place, and of course folding the body up into, so that's got a lot of surface area, which is kind of related to this, but internal surface area, lots of body cavities for things like that to occur. And then finally, this is the most sophisticated of all of these answers right here. How can we increase concentration differences? There are a number of different mechanisms, but the one that's a good example is a countercurrent exchange that we'll take a look at here. So this lecture will cover all three of these in various details while looking at different respiratory systems and how they do this. All right. So, as we look at different types of organisms, we see some, trends right here. So for unicellular organisms, they don't need to have any other particular equipment. Gas exchange, because we're talking about gas exchange or respiratory systems, just diffuse directly in. And very, very small multicellular organisms can also rely on direct diffusion across their surfaces. But most of the multicellular organisms, and certainly the complex ones that move around a lot, must have some additional mechanisms that do these three things, or some combination of these three things. So, for instance, insects, they've got an extensive tracheal system. So insects, living on land, have a series of air pathways leading through their body that reach deep into their body tissues and bring the outside environment very close to internal tissues that need gas exchange, muscles and things like that. Amphibians respire across their skin and, the lining of their mouth, if you're not aware of that. So their skin is a respiratory organ, as well as the lungs if they're on land, toads and frogs, or their gills, tadpoles, and some of the Sirenidae and things like that. They have gills as the adult phase. And so that's true for most organisms with a moist epithelium. The actual epithelium, the surface of their body, can be an exchange surface for gas exchange. The annelids, earthworms, and all of their relatives breathe, and they have gas exchange across their epithelium. Mollusks, aquatic arthropods, almost all the aquatic vertebrates use gills. Right, or all the aquatic vertebrates use gills. A gill, like a fish gill. So there's variations on this theme. Each group of organisms has their own peculiar details, but they all share the same basic, structural characteristics, and they function the same way. So when we're talking about gills, think fish gills, and you're getting the same thing done for these guys right here. And then finally, terrestrial vertebrates have this network of alveoli, in a thing called a lung, which is a bag with a whole bunch of small compartments inside it. So here we'll take a look at a gill. And I'm sorry about this note up here. There was an error in it originally that I fixed previously, and then I saved it that way. So don't worry about this right now. So what does a gill do? Well, first, by terrific folding all over the place, terrific folding and lots of small filaments, we get a huge amount of surface area packed into a very, very small structure. So the gills are, I mean, they're a prominent feature of a fish, and there's a bunch of them. There's 5 gill arches, as you can see right here. And they create a terrific surface area for gas exchange. So we're going to have a whole lot of blood flowing through the gills, which is why they're red, because there's lots of blood really close to the surface right here. And if you, hopefully you know that, what fish does either by moving forward in the water column or by gulping is it takes water in through its mouth, squirts it out through the gill arches, and then in the case of bony fish, there's a flap called an operculum, a bony sort of valve right here that opens up, and the water is ejected right here. Some fish have to swim forward all the time to ram water into their mouths to flow across the gills. That's true for almost all sharks. All right. So one modification is the great surface area right here. And then the other one is this countercurrent exchange right here. So when we look at, a tiny little bit of any one of these gills right here, we'll see that they are arranged so that water passes across, the exchange surface. And right under that exchange surface are a bunch of capillaries carrying blood, and the capillaries are running in the opposite direction as the water. So deoxygenated arterial blood comes in here and flows forward relative to the water flow, and then is picked up by an artery and running back, And that is called countercurrent, because the water is going in one direction, giving up something it's carrying, and the blood is running in the other direction, picking up, what it's picking up from the water and giving up what it's giving up to the water. And the idea is here the water has a low CO2 pressure and a high O2 pressure, and the blood coming in here has a high CO2 pressure and a low O2 pressure, and so we exchange both of those gases very effectively. All right. Let's take a closer look at that countercurrent. All right. So here's that same illustration before: blood running in this direction, water running in that direction. Excuse me. So if we look a little more closely, let's take a look at this one right here. So here's what's going on. Here's the blood coming in this direction with, the minimum amount of oxygen it's going to have. We'll just call it 0% right here, but you'll still have oxygen in your blood. And then as we get over here, we've picked up the maximum amount we can pick up, which is just a little bit less than the maximum volume in the seawater, or the maximum pressure of oxygen in the seawater coming in. And then that pressure of oxygen in the seawater falls all the way to the end. And even when we're at 20% or 15 or 10 or 5% concentration here at the end, the blood is coming in with an even lower amount of oxygen right here, so we can still effectively transfer oxygen to the blood all the way here. If we have blood and seawater running in the same direction, we're going to equilibrate at 50% concentration shared between the two fluids. That's no good. So we want this. In this drawing right here, we use the actual pressures of oxygen. So as we see right here, normally the blood coming in has a partial pressure of about 30 millimeters of oxygen right here. The seawater coming in has a partial pressure of about 150 millimeters of mercury of oxygen in here, and begins moving oxygen into the blood, even though the blood has a lot of oxygen already right here, and can continue effectively transferring oxygen to the blood even when the concentration is relatively low in the seawater, because the incoming blood has such a low concentration of oxygen. So this is the countercurrent, very efficient. If we average those two values, we split this value right here, 150 in half for concurrent flow, we'd end up with only 75 millimeters of oxygen in the blood leaving the gills, whereas here we get 135, much, much better. So that's mandatory. That has to happen. And you'll find that in almost any exchange surface anywhere doing anything. In your digestive system, food runs in this direction as you're absorbing things, and blood runs in this direction, picking up nutrients, just as an example. So it's even in your intestines, you have countercurrent exchanges for picking up things from the digestive tract and introducing them into the blood. It's pretty universal. So countercurrent, make sure you can talk about that. All right, so In terrestrial organisms, gills are replaced by an internal structure. The reason for that is to reduce water loss. A gill works really, really well. It works even better in air as long as it's really wet. But the problem is it's going to dry out, or you're going to lose huge amounts of water in order to keep it wet, and that's just not possible. There are things with gills that wander around on land, but the gills are packed in and hidden inside a partially enclosed or almost completely enclosed structure. There are some arachnid spiders that have gills inside a container with a small hole leading to the outside world. It's called a book lung, because the gill has been converted into what looks like a slightly spread open book of leaves, and so on and so forth. But anyway, terrestrial organisms, terrestrial vertebrates are going to have lungs instead of gills as a rule. So y'all all know this part right here. You know, the airway consists of these conducting tubes right here. There's no gas exchange occurring right here. All we're doing is getting, you know, the atmosphere in, or the air in the alveoli out with this exchange, with this distributing system right here. The trachea is the one big tube. All the other tubes that branch off the trachea are called bronchi. The first branches are simply the primary. The second branches are called the secondary. The third branches are tertiary, and so on and so forth. The only other term of importance is a bronchiole, which is the smallest and muscularly adjustable tube that leads into a cluster of alveoli. When we have asthma, it is due to spastic contraction of the smooth muscle of these bronchioles that closes off access to those gas exchange bags. That's the source of asthma. All right, a little bit about pressures. All right, a little bit about pressure. So, know this number. 170 millimeters of mercury is the defined pressure of the atmosphere at sea level. And that's the result of the column of air up above pressing down on the air right above the surface. The weight of all of that air above the bit of air directly over the sea yields a pressure of 760 millimeters of mercury. And, you know, this comes from there's a contraption, a device where we can measure pressure using mercury. In fact, I'll just show that to you right now. This is where that idea of millimeters of mercury comes from. So if you have a glass tube, can be anything, but a glass tube that's sealed and has a vacuum in it, so there's no air in it, and you put that into a petri dish full of mercury. The mercury is very heavy, it's very dense, you know, but it will be sucked up by this tube as a function of the pressure exerted down on the surface of this mercury right here, and that distance that it will rise in a standard, you know, size tube right here. I have no idea what the diameter is, but I'm sure it's a standard value to get this number. That's where the 760 millimeters of mercury goes. If you take this entire apparatus without taking it apart, you know, keep the vacuum here, and take it up to Mount Everest, the pressure goes down right here, the column of mercury goes down, and that's how we measure air pressure traditionally. That's where that idea comes from. All right, so that's a little bit about air pressure, and that's where that 760 millimeters Hg comes from. And we call that one atmosphere, very convenient. All right, so the atmosphere, of course, is a mixture of a bunch of gases. Please know these three. Nitrogen is 79% of the atmosphere. The bulk of the air that you're looking at is nitrogen, 79%. Oxygen is just shy of 21%, so we'll just call it 21% here. Here's a slightly more accurate number. 21% of that 760 millimeters. And CO2 in the atmosphere is very low. We get terribly worried about CO2 as a greenhouse gas, and that's because even though the concentration is very low, it has a profound effect on the temperature balance of our atmosphere. So increasing this number, you know, even a bit, has a real big effect on atmospheric behavior. But the key point right here is that the pressure is very, very low. Lots of oxygen in the atmosphere, we need it. Very little CO2 in the atmosphere, that's good, because we want to dump ours into the atmosphere from our body. And so here's the resulting partial pressures. And, you know, 760 times 0.79, 760 times 0.209, etc., yields these values right here. And if you're not familiar with that math, 79% is the same thing as 0.79. 760 times 0.79 must be a number smaller, because this is less than 1. Must be a number smaller than this, so that's where that 600 came from. I'll walk through it in your mind a couple of times, and it'll be comfy. So, importantly, 760 is sea level pressure. The pressure at the top of Mount Everest, as it says right here, at 5,364 meters, almost 18,000 feet, is 401 millimeters of mercury. That means the partial pressure of oxygen up on Mount Everest, sorry, is only 84 millimeters of mercury. Well, there's still oxygen there, so you might say, well, that seems pretty good. Well, there's the sort of partial pressure of oxygen we want in our blood going to our body tissues. We can tolerate this being a little bit lower, but not much lower. And we really can't, it's not going to drop below 30 millimeters of mercury under normal conditions. Falling below this in your venous supply is a really, really bad thing. So imagine that being your source of oxygen. You've really reduced by a terrific amount the amount of oxygen that you can actually distribute to the tissues in your body right here. And in fact, you and I, without training, are probably not going to be able to live up here. We've got to have an extra source of oxygen. So partial pressure of gases is terrifically important. All right. We want to take a look at some alternatives to, we're looking at evolutionary adaptations in respiratory systems. So here's what's considered generally to be the most efficient gas exchange system possessed of any animal. Birds. Fish gills work really, really well too. They have to be given quite a bit of credit because they share some features of the bird respiratory system, particularly what's called the one-way flow through the exchange surfaces, which maximizes counter-current exchange benefits. But birds really have it. And if they didn't have this respiratory system right here, they might still be able to fly, but they'd only be able to fly for a short bit of time until they got tired. Like you can run really fast for a bit, then you get tired and you can't run so fast. That's about where they'd be. The ability to fly, for hours or even days at a time is totally dependent on making all their muscular contractions fully aerobic by a terrific supply of oxygen, and that's what this system does right here. All right, and so what happens is they breathe air in through their trachea, and it doesn't immediately go to their lungs. It fills these hollow air sacs in the posterior part of their body. All right, and at the same time that they're breathing in and filling up these posterior air sacs where there's no gas exchange, they're just storage containers. They also, by the way, make the bird really, really light, because inside it's got lots of air pockets. At the same time those are filling, gases are leaving the lungs and going to these anterior air sacs right here. To these anterior air sacs right here. All right. And then on exhalation, both of these bags contract. The anterior air sacs move their air to the trachea, and that gets blown out of the bird's mouth, while the posterior air sacs, when they contract, blow their air into the lung. And so the long and short is of this thing going through a cycle right here. Air only runs in one direction through the lungs, from the posterior to the anterior. Now guess which way the blood flows in the lungs? Posterior to anterior or anterior to posterior, according to this drawing. I'll let you watch this video, and I strongly encourage you to do it, but I'm not going to record it just to save a little bit of time right here, since you can just pop out and look at that recording. All right. Okay, let's take a look at mammalian lungs. By comparison, we have what's called a bellows type lung. So there's this musculature right here, this diaphragm, plus the musculature of our rib cage allows us to expand the volume of this compartment. So the diaphragm comes down when it contracts, and that causes the lungs to want to expand to fill that space right there, because it's a sealed space. And if we open up our mouth, air can rush in and it can happen. Try closing your mouth and holding your nose closed and try and breathe in and feel the strain on your diaphragm. It'll get tense, but it can't contract because the lungs can't stretch, and so it's held up here, and you can't make anything happen. When you finally release your fingers from your nose, air can rush into the lungs. They can stretch as the diaphragm pulls down on that cavity there. So it's kind of a neat way in which we breathe. So the tracheal and bronchial tree just breaks and branches until it finally gets to the 3 lobes of the left lung and the 2 lobes of the right lung. Sorry, the 3 lobes of the right lung and the 2 lobes of the left lung. There's a heart in the way here. So 2 lobes on our left lung, most mammals. And the action area is where these bronchioles lead into the alveoli, which is a series of connected little spherical containers wrapped in a tight network of capillaries. And there's a very short distance from air here to blood in the capillaries, and that's where we're going to have our gas exchange. And as much as is possible, circulation right here follows countercurrent rules. It's not perfect because air rushes in and rushes back out in 2 different directions right here, but blood flow is optimized to take advantage of any little hint of countercurrent exchange activity here. So we're looking a little more closely at an alveolus and the capillaries surrounding it, and this is the gas exchange surface of your lungs right here. The inner lining of the alveolus is a whole bunch of epithelial cells, squamous epithelial cells, and some immune cells wandering around the surface here looking for whatever interests them, and some type 2 epithelial cells which produce the slippery mucus that's characteristic of your respiratory system. And then this tight net of capillaries surrounding it right here. And of course the arteries are coming in carrying deoxygenated blood, hence the blue coloration right here. When they collect the blood, it's full of oxygen. The veins leading away must be necessarily red. So color coding, you may have learned like in primary school, red means arteries and blue means veins. Not true. Red means high oxygen, blue means low oxygen. So in this case, arteries are blue and veins are red. In your systemic circulation, red, arteries are red because they're coming from the lungs and going to your tissues, and veins are blue because they're coming from your tissues and going to their lungs. So that's how that matches together. And here, of course, is our airway. We're going to have lots of mucus glands all over the place right here because mucus is continuously produced in your respiratory airway, one, to keep this from drying out, and also the mucus is continuously pulled up your airway by the cilia. This is a ciliated epithelium right here. And that mucus is continuously pulled all the way up the respiratory tree, all the way back up these branches, all the way up these branches, and all the way up the trachea. And normally you don't even notice it. You just continuously swallow it as it comes up to the top of the larynx right here and enters your pharynx, your throat. If you're producing excess mucus, that's where the urge to cough or what I just did and cleared my throat a little bit occurs because the excess mucus, it manifests as an irritant when there's a large blob of it right here. All right, lung structure and function. So just to reinforce what I said earlier, this is true for all animals with lungs. The lung is in a sealed container. All right, the volume of this container can't really change unless we can add some volume to it. And so what we do is we allow the volume to increase inside here by allowing air to go down the respiratory tree and into the lungs so that the lungs are allowed to expand if we apply a suction to it. How do we apply a suction? By pulling down on the diaphragm. This muscle is always arched upwards here. If it contracts, it wants to try and straighten out, which effectively pulls the floor of the thoracic cavity down, increasing the volume. At the same time, you contract muscles right here to lift your rib cage. Try at home doing a deep, deep breath in and notice that your rib cage lifts as well as your diaphragm trying to go down. How do you know your diaphragm went down? Your stomach pooches out anteriorly. It gets bigger. And that's called negative pressure ventilation. So we apply a vacuum to the lungs so that they naturally fill by air rushing in to counteract the vacuum applied by the musculature. And then we apply positive pressure by contracting the skeletal muscles here and relaxing this, and contracting our abdominal muscles to shove our liver and stomach up. And that squeezes, compresses this pathway here, and if our airway is open, air will rush out the airway and out. So that is positive pressure exhalation, negative pressure inhalation. All right. A little bit about the way your lungs work. So you may have heard these terms, and I'll pull up a graph here and show it to you in just a second, but tidal volume and vital capacity. So tidal volume refers to the lungs the air that you're moving in and out of your lungs under normal conditions at rest, like me sitting here, although talking tends to make me take deeper breaths in between bits of speech. But it's going to be somewhere between 1 3/4 of a liter per breath, per person. Vital capacity is the total amount of air that you're capable of expiring from your lungs, and therefore, if you've totally emptied your lungs, the absolute maximum amount of air that you can suck in. That's called your vital capacity, which is a good measure of your cardiovascular health, or at least the the cardiopulmonary portion of it. And for somebody like me, historically, last time I had it checked, it was just under 6 liters. That was my vital capacity. As I age, it's going to go down a little bit because of natural loss of flexibility and elasticity in my lungs. When I was younger, it was probably a bit higher. All right. Oh, let me show you the graph. All right, sorry, this is a little bit blurry, but don't worry about it. I think we can see it just fine. This right here, maximum inspiration, maximum expiration right here. And this is maximum expiration. So this is the volume we can get to. That is your vital capacity, this distance right here. That's the vital capacity, the amount of air that you can move in and out of your lungs. Why we're interested in that and why this is different here is if I blow my lungs out completely, squeeze out every bit of air I can, my trachea and my 2 primary bronchi and all the distributing bronchi, and to a small amount all the alveoli still have air in them. So this is that dead residual volume. We never can blow this all out. So when I take a breath of air in from the outside world, full of oxygen, I mix it with this residual volume in here that has low oxygen and high CO2. It's a limitation of the bellows lung. Anyway, that's what we mean by vital capacity. So here's everything I can breathe in or out. If I'm sitting and resting, this is my tidal volume right here, the breaths I'm taking in and out when I'm barely doing anything. And then you decide consciously to take a deep breath, fill your lungs all the way out, blow everything you can out, that's all the way down. You have just demonstrated your vital capacity. All right, so now let's go on and take a look down at the more molecular level of gas exchange and respiration. So we're taking a look at hemoglobin now. So in you and in all vertebrates, we're going to have red blood cells that are specialized cells optimized for just gas transport. And they are primarily full, after you account for the liquid in there. Water may be, or plasma may, cytoplasm may be the first component, but, by far the next most abundant thing in there is going to be hemoglobin molecules. And that's about all they carry, just a heck of a lot of these dissolved in cytoplasm of a red blood cell. And the hemoglobin molecule, hopefully you can recognize it right here, is in fact 4 proteins, 1 alpha, 2 alphas, a beta, and another beta. So there's 2 betas and 2 alphas at cross angles to each other right here. And that's a globular protein. And then in it is a heme group, which is a carbon, a 5-ring, 5, 6, we'll look, a multi-ring carbon molecule right here with a whole bunch of nitrogens on it. And then smack dead in the center is going to be an iron atom. And that's the reactive center for the heme group. And that's where we get hemoglobin. The heme group is this multi-ring carbon molecule right here. The globulin is the big protein that is embedded in hemoglobin. And this heme group right here is recovered in your spleen and also in your liver from dead red blood cells from exposed hemoglobin. And the heme group without the iron and missing the globular protein is modified to make bile in your liver. And that's the source of bile, is recovered and reused heme groups from hemoglobin. All right, let's see if we can take a look at the heme group right here. So we'll just take a peek at this really quickly. Here's a 4-unit hemoglobin molecule, and right here they've got the 2 betas on the same side and the 2 alphas on this side. And I see this error particular. To the best of my knowledge, it's alpha, alpha, beta, beta. They're kind of an X-shaped arrangement, but it doesn't really matter too much right now. And here's the heme group suspended in the center right here. These are covalent bonds right here holding this into the protein. And here's what it looks like. So it's a 1-ring, 2-ring, 3-ring, 4, 5, 6, 7. It's 7 rings, or 4 rings, and then sort of skeletal parts of a ring right here, and then a centrally bound iron atom right in the middle right here, and you notice the 4 nitrogens around it. So remove the iron and get rid of the globular proteins, and the remainder of this is converted into bile, because it's As you can see, it's it's got a lot of insoluble parts which will hang on to lipids, but it does have some soluble parts in just a couple of key locations, these acid groups right here, that allow it to dissolve in water. So it can hold on to oils and water at the same time, which is what makes bile bile. So anyway, a little bit about the heme group right there. Carrying on, let's talk about the behavior of hemoglobin. So hemoglobin binds to oxygen, and its ability to bind to oxygen, which is called its affinity, high affinity, a strong bonding, low affinity, a weak bonding, is changed by pH and temperature. And as we'll see, it kind of works in our favor right here, okay? The shift in O2's ability to hang on to oxygen, hemoglobin's ability to hold on to oxygen due to pH is referred to as the Bohr shift. A researcher named something Bohr, Dutch or German or somebody like that, described this effect, and so it's named after that individual. But it shows the change in affinity for oxygen. Let's go ahead and take a look at the graph right here. Okay, so here's the partial pressure of oxygen. Anywhere between nothing and 100 millimeters of mercury that we might encounter somewhere, somewhere in our respiratory path. Numbers like this we're going to see up near our airways. Numbers like this we're going to see deep down in tissues that are using oxygen like crazy. And here's the percent saturation, how much oxygen the hemoglobin can hold on to under these different pressures right here, this being 100% being the maximum, and you can see this curve is asymptotic approaching 100% right here. Well, what happens if we lower the pH, if we make the fluid that the hemoglobin is in more acidic, or if we increase, which is a result of increasing the amount of CO2, higher CO2, more CO2 dissolves in water, that becomes carbonic acid, which lowers the pH. Also, at higher temperature, hemoglobin has a harder time holding on to oxygen, so we lower this curve. It's lower. We're not holding as much oxygen under these conditions right here. Conversely, if we raise the pH, we lower CO2 concentration and we raise pH. Normally this comes up when CO2 goes down. Get rid of CO2, that gets rid of carbonic acid, that rises pH in our blood. And that's the way it works in our blood. The pH in our blood is a function of how much CO2 is dissolved in it. And if we lower temperature, all of those things will cause the ability of hemoglobin to bind to oxygen to go up. Well, it's kind of neat. In our lungs, we encounter this set of conditions right here. Relatively low temperature. Why? We just breathed in cold outside air into our lungs. Even though our core temperature is high, momentarily, and if we're breathing rapidly, pretty much continuously, the temperature in our lungs is low. We're also losing CO2 to the atmosphere, so the pH tends to go up right here. So hemoglobin is extra good at binding to oxygen in our lungs, just like we want. In the center of your thigh muscles while you're running is where you're going to get the most extreme set of these conditions right here, where you're going to have very high CO2 concentrations in those tissues and therefore a very low pH. Think of all the lactic acid also, coming out of your legs if you're really running hard, and a very high temperature because you're, emitting a huge amount of lost heat during cellular respiration right here, and so generating a very high temperature. Well, hemoglobin loses its ability to hang on to oxygen, so it preferentially lets go of oxygen down here deep in our very, very intensely working tissues, and it preferentially grabs oxygen in the conditions of our lungs. That's the Bohr curve in a nutshell there, and that's pretty neat. All right, just about the last material right here. We'll just scan ahead. Yeah, just a couple of, whoops. Didn't expect that. Let's make that happen again. There we go. Last couple of slides right here. We're taking a look at the transport of carbon dioxide in your blood, which is a little more complicated than the transport of oxygen. And the details are important enough at this level of your education, we want to talk about it a bit. So in your lungs, in your tissues, you're going to have high CO2 levels. So, all the CO2 originates in your tissues. So it's dissolved in cytoplasm of tissues. It's dissolved in extracellular fluids. It's dissolved in the plasma of your blood. All right. But when it enters a red blood cell, which it does, the CO2 is combined with water to make hydrochloric acid. That's this right here. And that hydrochloric acid decomposes automatically. It becomes a real acid when it lets go of its hydrogen ion, and this goes into solution. So there's the acidic proton that makes this an acid. And this part is a base right here. So there's a base remaining right here, but this is the thing that contributes dramatically to the pH right here. Well, the bicarbonate is then going to be transported out of the red blood cell, and it's negatively charged, so that's going to induce an electrochemical gradient, which preferentially allows chloride to go into the red blood cells. And so the HCO3 right here is this bicarbonate that provides the primary pile of bicarbonate, which is a buffer now. Without the acidic proton nearby, this acts as a buffer to keep pH relatively neutral in the blood, which is what we do. I mean, we're wandering back and forth between 7.5 and 7.3 and 6.7 or 8 or something like that. Very small shifts in pH are a big deal right here. And when we get to the lungs right here, where the partial pressure of CO2 is really low in the alveoli, what happens is that bicarbonate, CO2 starts dissolving out of the cytoplasm of the red blood cells and into the plasma of the blood, and from the plasma into the alveoli. So the CO2 is leaving, which drives all of this reaction. And this is an example of buffer chemistry reaction because it's easily reversible. The reaction runs in the other direction. So bicarbonate is combined with a hydrogen ion back into carbonic acid, dissociated into water, or broken down into water and CO2. And there's an enzyme right here that catalyzes the reaction called carbonic anhydrase. And it catalyzes this reaction in either direction here. And so we lose CO2, and the buffer returns as a means of moving CO2 back into the red blood cells and back out into the alveoli. And chloride ions are shifted back into the plasma of the blood. So that's what we do. One of the things we do with the chloride from sodium chloride table salt. We use that right here. So let's know these numbers here. All right. 8% of the CO2 in blood is dissolved in the plasma. So that's the CO2 right here. It's just sitting as CO2 dissolved in water, in water of blood, in plasma. 20% of that CO2 in its molecular form of CO2 is bound to the hemoglobin molecules inside the red blood cells. The same way oxygen is carried, but only some CO2 is carried that way. The remaining, about 72%, is converted through this chemistry here into the bicarbonate system, and that's how the bulk of our CO2 is carried. What's important about this is that we've got CO2 trapped in a new molecule, bicarbonate. We've got CO2 in its original form, and we've got CO2 complex to a protein. If all of this was just CO2, 100% of the CO2 would be a smaller volume than the CO2 we can carry when we carry it in these three distinctly different chemical forms. So the fact that we're carrying CO2 dissolved directly into liquid, CO2 bound to hemoglobin, which means it doesn't count as this. It doesn't count towards the solubility of CO2 anymore because it's bound to hemoglobin. It's a new chemical. And the same with this bicarbonate. This bicarbonate doesn't have anything to do with the hemoglobin or the dissolved CO2 right here. We've got three ways in which we can store CO2 so we can carry a lot more. That's the important thing. When we look at the movement of oxygen in blood, there's a similar, although slightly less dramatic thing. O2 is dissolved in the plasma of the blood. You do carry some oxygen that way, 10%. You carry oxygen dissolved in the cytoplasm of the red blood cells, another 5 to 10%. But then the overwhelming majority of your oxygen, over 80%, is carried by hemoglobin itself. It's attached to the hemoglobin molecules in the red blood cells. But the importance of there with oxygen too is that we've got 1, 2, 3 different compartments / forms that we can store oxygen as, and that increases our ability to carry oxygen by the blood as well, which is an argument for having blood that has cells dedicated to gas transport. All right, a little bit about CO2 and O2 measuring and respiration. So ventilation is the act of expanding and decreasing the volume of your chest cavity for pulling air in and out of your lungs. That's ventilation. So when we say hypoventilation, we're not doing enough of that, breathing. Hyperventilation, too much breathing. But ventilation is the proper term for breathing. Respiration, we have to be specific. Respiration normally talks about these ideas. This is exterior respiration between the alveoli and the blood. This is interior respiration between blood and body tissues. And then cellular respiration, of course, is what happens inside cells using CO2 and using O2 and producing CO2 as a byproduct. That's cellular respiration. This is internal respiration. This is external respiration. Now we're talking about ventilation, which is what you and I call breathing. All right. So normally what you measure in order to regulate your breath, if you hold your breath, the thing that causes you to get a stronger and stronger urge to take a breath is actually measuring the concentration of CO2 in your blood, because what's happening is you've got cells that very accurately measure both the CO2 directly, but even more accurately measure the pH of blood. And CO2, as its pressure increases in the blood, you inevitably create that carbonic acid, and that releases hydrogen ions into solution, which is what we measure here. So when you hold your breath and you finally give up and you have to take a breath, it was really the change in pH that drove you to do that. You never ran out of oxygen. We never want that to happen. We want our oxygen levels, I'm going to make you dizzy by running back through the PowerPoint right here. We want to keep our oxygen levels pretty high. Where is it? Oh gosh, it was in the countercurrent thing here. There we go. In this picture here, we don't want to get our partial pressure of oxygen in us. It's going to be about 40 millimeters of mercury of oxygen in venous blood, deoxygenated blood coming back, to our lung. That's as low as we ever want to get it. 30 is really low when we're super, aggressively exercising. The point is, we've still got enough oxygen left in our lungs, in our blood, to power ourselves for, a few more revolutions, a few more cycles before we seriously run out of O2. So the point is, to regulate our breathing, we don't normally measure oxygen, we measure CO2. Now what will happen is if we lose track of that signal, so the brain isn't signaled to initiate a breath, the person feels like they can't breathe and all of these sorts of things, they start hyperventilating. We've gotten into a sort of a short circuit where we've lost our ability to regulate based on CO2 measuring right here. All right, so just to summarize regulation or detection and regulation of breathing, that is primarily done by measuring blood pH levels. And you've got some neurons that are chemosensitive. They have receptors that measure the pH literally. They use the same structure that we use to taste sour things on our tongue. It's the very same sort of receptors. They're just plugged into neurons directly, and they're not measuring sourness. They're measuring pH literally. And those are in the aortic arch. The main aorta, as it comes up out of the heart, does a loop and then goes back down. Up in that upper arch, we've got some measuring nodes right there where we got some of these neurons. And then also in the carotid arteries, where the external carotid that goes to our skull and scalp separates from the internal carotid that goes into our brain. Right there at that juncture, we also have some pH measuring cells. And that's the primary thing that we use to control respiration. And as it says right here, we also have in our central nervous system chemoreceptors that measure the pH of cerebrospinal fluid. And if that becomes more acidic, that triggers more rapid breathing and deeper breathing, ideally. All right, just to wrap it up, I talked to you a little bit about asthma previously. So there's a number of things that trigger asthma, but what's particularly happening is that the smooth muscle of the terminal bronchi, just before you get to the alveolar clusters, contracts, and that leads to restricted breathing. And if your metabolic demands are really, really high and the constriction is really, really extreme, it can actually lead to suffocation and death. So that's why people can actually die of asthma. There's any number of triggers. Classically, it's an external stimulus like an allergen or something like that, but there are also some internal malfunctions. But it's a hypersensitivity of your immune system, and histamine causes smooth muscle contraction of your airways there. The defensive reaction is meant to protect the airways from a harmful substance, but of course, we can't tolerate too much of that. Another pretty important respiratory disease that we want to characterize right here, because measuring vital capacity, residual airspace, and tidal volume is one of the easy ways of diagnosing whether somebody has emphysema right here. And this is the degenerative disease where the lungs become less elastic. This inevitably happens as you get older, but it's a small amount, 10% or something like that. But excessive loss of elasticity due to the breaking of walls between alveoli, so that your lung becomes, larger but fewer alveoli, and therefore, bigger and bigger compartments with less surface area collectively, is the phenomenon of emphysema. And two things: you get a lot of scar tissue in your lungs, so that scar tissue is replacing elastic connective tissue, so your lung becomes less pliant, so it can't stretch as much during inhalation, so you can't take a deep breath as well, and you lose surface area inside your lungs, so you have less surface area for gas exchange to occur. And as you can see, there's a number of environmental materials that contribute to that. Smoking, extreme air pollution, particulate matter in the air particularly, also low pH, like acidic. Say if you walk into a very acidic mist, you're going to get some lung damage, and a whole lot of that over a long period of time will lead to emphysema-like characteristics. So a little bit about lung disease, just as a finale right here. One last bit, talking about lung cancer right here. And like so many cancers in your body, lung cancer is typically a cancer of the epithelium. So I'm just pointing at this dissected out lung, which looks pretty bad. But the cancer is typically the epithelial surface lining the inside of the lung. Epithelia are one of your most rapidly reproduced cell types, along with the cells that make up your red and white blood cells. And there's another place where we get a heck of a lot of cancer. So most of our cancers are epithelial tissues, which are inevitably dividing rapidly and being replaced rapidly. In your lungs, if there's a lot of environmental stress or a lot of disease or whatever, you'll tend to have an elevated rate of epithelial cell replacement in that tissue, and that increases the chance for a mutation to lead to uncontrolled division of cells, which is the basis for all cancers. And then, of course, if we have environmental components, like a lot of pollutants, stuff coming from cigarettes and things like that, carry toxic compounds, carcinogenic compounds that tend to drive genetic mutations, damage to genes, breakage of DNA, and misrepair of that DNA, so that we lose the function of a controlling gene. That's generally the 1-2 punch of a cancer event. So, a little bit about those. You should be able to answer a question or two about emphysema and asthma, lung disease. It's not a major thrust of the lecture right here. We're more concerned with the fundamentals of lung function and lung structure. But that concludes this lecture on the respiratory system, gas exchange in humans and invertebrates particularly, and also look at the evolution of respiratory systems in animals.